Hello everyone. Today we're going to be talking about meiosis. How are sperms and eggs made? For the production of a baby, you need the sperm of a male and egg of a woman. When sperm travels and gets into the egg, an embryo, which later will be a baby, a human being, is produced. But where does the genetic information come from? The sperm of male and egg of female carry the genetic information to create an embryo. For the production of a human embryo, 46 chromosomes are needed, 23 from sperm and 23 from egg. Chromosomes carry the genetic information and have identical chromatids. So, how are gametes made? Meiosis, a reduction division of chromosomes, occurs during this process. But before getting into meiosis, we need to know what mitosis is. Mitosis is a type of cell division that results in two daughter cells, each having the same number and kind of chromosomes as the parent nucleus, typical of ordinary tissue growth. But don't get confused. Mitosis and meiosis are not the same thing. They are spelled differently and they follow the same cycle, but meiosis does it twice. Before mitosis and meiosis occur, interphase happens before process starts. Interphase is a process where the cell is growing, replicating its DNA, and making sure all the cell functions work.
The first stage of meiosis is prophase 1. Here, the chromosomes begin to condense and they line up with their homologous pairs. This process is called synapsis. A homologous pair is a pair of chromosomes that are similar in size and contain the same types of genes in the same locations. Then, crossing over occurs. This is when the homologous pairs transfer their genetic information with each other. The next stage is metaphase 1. Here, the chromosomes line up in the middle of the cell. Once they're organized in the middle, they can orient themselves however they want. This is called independent assortment. Then, once the centrosomes are on each side of the cell, the spindles attach themselves to the centromeres that are in the chromosomes. The centrosomes are an organelle from which spindle fibers develop in cell division. Then comes anaphase 1. This is when the spindles pull apart the homologous pairs and they go to either side of the cell. The last stage of meiosis 1 is telophase 1. Here, the cells begin to form new nuclei on each side. Then, the rest of the cell divides in a process called cytokinesis. So in the end, we end up with two new cells. Now begins meiosis 2. The first stage of meiosis 2 is prophase 2. Not much happens in this stage and there's no crossing over like there was in prophase 1. In metaphase 2, the chromosomes are going to line up again in the middle and the spindles are going to attach themselves to the chromosomes. In anaphase 2, the spindles are also going to pull apart the chromosomes, but this time the chromosomes will divide into chromatids. Lastly, in telophase 2, new nuclei begin to form again, and then cytokinesis happens to create 4 new cells. In this table, we can clearly see how the amount of chromosomes and chromatids changes. Before interphase happens, there were 46 chromosomes and 46 chromatids. After interphase, 46 chromosomes and 92 chromatids. After meiosis 1, 23 chromosomes and 46 chromatids. And lastly, after meiosis 2, 23 chromosomes and 23 chromatids.